welcome everyone. Welcome to the Impact Scholar Seminar presentations. And I'm going to give just a brief introduction to the Impact Scholars because we have people from outside. So the Impact Scholar is a 6-month initiative that brings together NeuroMatch and Climate Match students to collaborate in international teams on research projects that began during our July courses. And the program culminates with an open access micro-publication that will be shared on Zenodo, a public summary, and seminar presentation. And today we're actually going to hear some of the seminar presentations. So there are some instructions that I would like to share about today, because we have actually presentations from 4 groups. As a reminder, each group will have 15 minutes to present, and this will be followed by 5 minutes of questions. And at the end, after all the presentations, if there are still questions, there will be additional time for other questions. So at the moment, our first presentation will be from Mind Mappers Group from Computational Neuroscience. And they're going to present a project titled Social Cognition in Autism, Predicting Task-Based Neural Activity from Resting State Connectivity. So guys, I'll now hand the stage over to you. And thank you everyone for joining us today.
Thank you. Just a moment, I'll share my screen.
Sure.
Are you able to see my screen?
Yes.
Cool. Right. So can you hear me? Hello, everyone. Today, I'll be discussing my topic, which I completed with my team, Social Cognition in Autism, Predicting Task-Based Neural Activity from Resting State Connectivity. Starting with what is social cognition? It is understanding, predicting others' thoughts and emotions. It relies on a distributed social brain network. And in ASD, it is marked by disruptive connectivity within the social brain network, altered DMN connectivity, and atypical posterior STS activity during mental state attribution tasks. Neuroimaging insights reveal that in resting state fMRI, there is underconnectivity in DMN, overconnectivity in sensory or salience networks that correlate with social deficits and repetitive behaviors. Task-based fMRI is challenging in ASD due to motor artifacts, sensory sensitivities, and symptom heterogeneity. Our research question is, can we understand social processing in autism without using a single social task in fMRI scanner? So task fMRI is often unavailable and unreliable, especially in clinical populations. Resting state fMRI can accurately predict task-evoked activity as demonstrated in schizophrenia-related researches and neurological disorders. Machine learning approaches like BrainSurf CNN has applied transfer learning to predict task functional connectivity from resting state. We used activity flow mapping. We used activity flow mapping for predictive modeling. Activity flow mapping helps visualize how neural activity in a region influences others through an intrinsic network. With respect to ASD, few studies have integrated resting state with task activations. And predictive modeling offers a non-invasive, scalable method to estimate social cognition-related brain activity, even without task fMRI. The aim of our study is to explore activity flow mapping to predict ASD task activations using resting state FCs, leveraging task related data as a reference to model ASD specific connectivity. We utilized 2 data sets for our research. First is the HCP young adult dataset pre-processed with Glasser parcellation by Neuromatch Academy, which consisted of 339 subjects with resting state fMRI and social cognition task data. We also utilized a B-Day dataset, which was pre-processed using Craddock parcellation, consisting of 408 ASD subjects from a B-Day, but only 150 were retained due to data inconsistencies. The challenges we faced during our research was that we could not fully process the HCP dataset, as processing the 1200 subject dataset was time consuming and challenging. The Atlas conversions from Glasser to Craddock in the two datasets we incorporated and applying transfer learning was also challenging. In the Abide dataset, we found that subjects had varying ROIs, so only 150 subjects were valid for us to use. The things that were helpful during our journey was finding an activity flow mapping template prepared by Cole Lab, which made it easier for us to adapt the methodology. Having access to pre-processed NMA HCP dataset, which eased our analysis further, and developing an Atlas conversion pipeline for converting Glasser parcellation to Craddock parcellation in HCP dataset. This was done to facilitate comparison, which led to uniform feature representation and interoperability. So in the first step, we utilized the HCP dataset, wherein we used the resting state data and task data from HCP in Glasser parcellation, converted it to Craddock parcellation for alignment. Thereafter, we computed the multi-regression functional connectivity on resting state data. And then task activations were predicted using activity flow mapping. Further, the weights from this prediction were stored for transfer learning. In the third step, we utilized the Abide dataset, wherein the resting state data, which was already present in Craddock parcellation, was utilized. The HCP learned weights were applied to the Abide functional connectivity data, and the predicted Abide task activations were generated, keeping in mind no true task data was available in the Abide dataset. Multi-regression analysis was done, and activity flow mapping was completed to visualize predicted activations. Finally, the validation process included computing group-wise functional connectivity differences in Abide, computing the multiple regression to evaluate prediction differences, and also computing task activations and correlating them with ADI-R autism severity scores. Finally, visualizing all the data.
We successfully mapped activity flow to predict social task brain activity in individuals with autism. We normalized the predictive activity, showing the variability across 150 subjects.
So from this image, it is a predicted task evoked brain activity in ASD. We can find that there are some strong activation in the regions like parahippocampal regions and fusiform gyrus, and they are all related to specific functions like spatial memory, visual processing, and self-referential thoughts, especially the posterior cingulate cortex, and not in a deformal network. And these functions are partially relevant to the social cognition tasks, so it might verify that the F1 model could predict the social task brain activity in ASD.
To visualize the differences in brain activity, we used direct subtraction, as can be shown in this figure. There were notable deviations despite there being no significant differences. The largest difference was found in the medial prefrontal cortex, posterior cingulate cortex, and parts of subcortical structures. These are associated with the default mode network and the salience network, which, as mentioned earlier, have been known to show abnormalities in autism individuals. This is in line with neurobiological data.
So, as mentioned, there are no statistically significant difference found between the comparison between the ASD and the TD populations. It might indicate that the functional architecture of the social cognition task is preserved. This architecture is similar between ASD and TD. And the raw activation comparison showing that the activation network are related those activation network are related to the social connection. So the potential brain activation may really resulting from the experimental stimulus. And the previous research also found alternation in this network in ASD. So we may say that an Airflow model did a relatively accurate prediction. But on the other hand, these evidence are all from the resting state rather than during the task scanning. So we put a question mark here and we need to draw this conclusion later.
The B-Day dataset provided us with the ADIR scores, which are used to assess and diagnose ASD through several categories. To validate our model, we applied Pearson correlations between predicted activation levels and the qualitative abnormalities in communication subscore in ADIR. We found that there was a positive correlation between region 195 and the ADIR verbal scores, and this is related to higher ASD symptom severity and communication. This is in line with existing research. Previous studies suggest that there is greater right superior temporal gyrus activation in social tasks, and this is positively correlated with ADIR communication scores. However, in our data, this was not evident or replicable. It's important to note that despite statistical significance, the correlations are still relatively weak. This could be due to several limitations of activity flow mapping, which we will delve into throughout our discussion.
So there are some significant correlation found in the activated brain regions and the ADIR score, and some previous funding have similar outcome, like the region 195, the lingual and calcrane region positively correlated with the ADIR score in our research, and some other research found that they are actually linked to the sensory differences in the ASD, but rather than the communication. Moreover, the inferior frontal gyrus, which is negatively correlated to the ADIR verbal score in our research, does find association with the social impairment in ASD. However, this evidence is a structural evidence, but not a functional. So further investigation is needed. So we also think about why we did not replicate the previous results. It might be because that the demographic characteristics in two studies are various, and there is a huge heterogeneity in ASD population. Different individuals have different level of symptoms. And last but not least, the Airflow mapping may perhaps not performing at the optimal level. Although there are some inconsistent outcomes, the predictions does show some activation related to the social cognition task and ASD-related brain activity. So now we can conclude that the Airflow modeling is potentially able to predict the task activity brain data, but the accuracy needs to be further verified.
We assumed in our investigation that neurotypical task activations can be generalized to ASD populations, despite ASD brains exhibiting distinct functional connectivity patterns. To address this, we applied our transfer learning approach, which was effective to some extent. Activity flow relies on direct comparison between task and resting state data, but as mentioned earlier, the B-Day dataset lacks task data. Furthermore, there hasn't been a lot of task data specifically related to social cognition through neuroimaging in autism. The activity flow mapping is a theoretical framework, and it does not inherently have any predictive modeling capabilities. Our study that used transfer learning has not been done before using activity flow mapping. Transfer learning using the weighted vectors we used may not have been optimal. For example, it might have been more effective to compare between two autism data sets to predict task activity rather than using neurotypical task activity to predict autism. And this has not been done before, so testing the viability of this method is difficult. Previous studies have shown that it is possible to use such a method. However, it has not been done or verified through activity flow mapping, which weakens the predictive power of the model. Activity floor mapping itself is limited and it's sensitive to individual differences, so it might be better to use machine learning techniques in addition to this method for greater generalization in the future. Despite limitations, our investigation did have many strengths. Unlike traditional connectivity models that rely on static correlations, activity flow captures the directionality of brain activity, illustrating how one region influences another through functional pathways. This perspective aligns with our understanding of the brain as a dynamic interactive system, rather than a collection of isolated regions. Though theoretical and not based on machine learning, this simplicity enables an initial exploration of brain network dynamics, which can inform the development of more complex predictive models. It is difficult to obtain task data for ASD due to it being a time-consuming and money-intensive process, accessibility challenges, and participant burden. Using predictive modeling, we can generalize trends that may be consistent in all ASD participants despite individual differences without having to manually collect imaging data. Our method may allow researchers to simply use resting data to understand the neurobiology of task states, which would make research more efficient. This can be a preliminary step to be used alone or in conjunction with other methods for obtaining task-based neural activity. Ultimately, this method can be used as a stepping stone to understand and predict individual variability, leading to earlier diagnosis and personalized interventions for ASD patients.